All right. Uh, so we were talking about a sclera here, which once again is the outermost layer of the eyeball. And it's a white part of the eye, and it can get infected and it might turn pink. And people with jaundice, for example, they might have a little bit of like a yellow pigment that starts to build up in the eye as well. And you can see that the sclera over here provides attachment sites for various muscles that would control the movement of the eyeball. You have one, two, three, four, five different muscles over here. The top one helps you roll the eye up, and the bottom rolls it down. It's pretty self-explanatory, but you have one over here is called the inferior oblique. It's kind of on a diagonal there, and it allows you to roll the eyeball. Okay. The front part of the eye is going to become the cornea, and that is the convex structure you can see right here, which allows you to capture light coming in from a wide angle. Hold on, guys. It's not going forward. I don't know why. Give me a second. Okay, this is super weird. Hopefully, it's gonna be okay now. All right, moving on. There's this news article I saw a while back where people were doing tattoos on their sclera. And if that sounds like a bad idea, it really is. What happened was there have been incidents where it leads to severe infection afterwards, and people actually lose visions as a result of the procedure. And since then, in the U.S., the Washington state actually tried to ban this practice, and Canada, of course, has already banned it. It's not a good thing to do. Okay, so the layer that's underneath the sclera is the choroid, and the choroid, as we mentioned during the labeling, contains blood vessels to nourish the eye. And at the front of the choroid, that's where you will have the tiny opening to allow light to come in. And that opening is called the pupil. You can see the black circle in the middle. That's the pupil. And that is surrounded by a muscular layer called the iris. And the iris can have different color depending on the genetic makeup of the person. Hazel eye, chestnut brown, so on and so forth. And it's not just for creating the color of the eye. The iris actually is a muscle that is controlled by your autonomic nervous system. So just to refresh your memory a little bit, your ANS actually is separated into the uh, sympathetic and the parasympathetic system. And the sympathetic system is for fight or flight response, and it will actually dilate your pupil. Whereas for parasympathetic system, it is going to constrict pupil, make it smaller. Now, another interesting thing is the two pupil size are independent of each other. If you're wearing an eye patch, for example, it's pos and you're outside, your right eye is going to have a smaller pupil compared to your left eye, since your left eye is in the dark, right? So they don't have to be both dilated or both, constricted at the same time. And by changing the size of the pupil, it allows the right amount of light coming in so you can see things clearly. It's the same thing with the camera, right? If you let too much light in, then your photo is going to be overexposed, and you can't really see things. Everything is just very, very bright and like a white hue. Um, Again, if you go to the eye doctor, sometimes they give you these eye drops to dilate your pupil, so that they can see the inside of the eye better. And afterwards, they tell you like not to drive and you need to wear sunglasses because the pupil is going to stay dilated for some time and you might not be able to see clearly, right? In fact, I remember going to the eye doctor once. And after dilation, I was not able to read for the rest of the day. And I actually had an exam the next day, so I wasn't able to study at all, because dilated pupil does not allow me to focus the reading as much. So that's the core layer with all the other structures associated with it. If we go one layer deeper, then we have the retina. Okay, so the retina um, is where you will find photoreceptors. I already told you rod cells are for night vision and peripheral vision, whereas cone cells are needed to distinguish colors. This is an actual pictures of the cone cells and the rod cells, and they are basically named after their shape. The cones are kind of, triangular in shape, and the rods are more cylindrical in shape. And these things are gonna make connections with various sensory neurons and send the information to the brain. So we only have three types of cone cells: the red, the green, and the blue over here. And when you um, stimulate more than one of these cone cells at a time, that's how we register different types of colors. When you register red and green together, then you see the color yellow. Now, I want to clarify that these colors that we are mentioning, these are light color. Light color is not the same as pigment color. Like if you think about the color that you use to create a painting, if you mix red and green together, you are not gonna get yellow. Okay, if you mix red and green, you actually get brown, I believe, right? But these are light color that we're talking about, right? So you have to understand that there is a difference. So red and green together makes yellow. Red and blue together makes magenta, and then blue and green together makes this color called cyan. And if they're all activated at the same time, then you actually get white. Sometimes you can have stronger red and weaker green stimulation. Then the yellow will shift a little bit towards the red, creating an orange color. All right, so it's just a combination of red, green, and blue, and the intensity of each of the colors that will create, all the different colors that we see. Now, I am going to try to explain this a little bit further for you, and try to follow along with the screen and with this activity next. It might help you understand a little bit more. So remember, there are only three cones in the body: red, green, and blue. And to see white, you need all three. So what I'm going to do is I'm going to switch to the next screen here. And I want you to stare at the red screen as I'm explaining things to you, okay? To see this red screen here in front of you, to see this red color, only your red cone cells are being stimulated. All these red pixels from the screen is going to enter your eyeball. It's going to go and hit the cones that are located on your fovea. Okay, and it's only going to stimulate the red cones. Right? The blue and the green are doing absolutely nothing right now, right? Because these red color, it's actually wavelengths, and this particular wavelength will only be able to trigger your red cone cells. Now I'm going to switch to the next screen, and I want you to blink a couple times, okay? Tell me, what color do you see when you blink on this screen? What color are you detecting with your eyes? You see white? Anyone else not see white? Or you all saw white? I mean, it's white now. It's definitely white now. But a moment ago, when you were blinking your eyes, yeah, there was a bit of blue, right? That's right. So what happened was this is a white screen. To see the white screen, you need all three cone cells, the red, the green, and the blue. However, by staring at the red screen for a long time, and if you had been staring at it hard, the red cone cells are tired. Okay. So when you switch to a white screen, the green and the blue are able to activate, but the red is a little bit exhausted, so it comes off a little bit weaker. And that's why you saw the cyan color on the white screen. Does that make any sense? Okay, maybe I try another question with. I think I have a good practice question coming up anyways, okay? But let's try one here. Okay, let's say you are staring at a green screen for a long time, green screen, and then I switch to a yellow screen. What would you see on the yellow screen? Eventually you will see yellow, but on the yellow screen, after the green screen, what color will you see? Okay. Red is correct. Okay. Because to see yellow, you need red and green. But if the green is tired out on the yellow screen, only the red will be working. And so you will see you will see red on a yellow screen until you blink a couple more times, and then the green receptors recovered, and then you will see yellow again. Okay, so here is based on that kind of understanding, uh, these kind of optical illusions are very common. Right? I don't know if you've tried these before. What you need to do is you will stare at the blue dots. Okay, so when you're staring at the blue dots, this picture with all the wrong colors doesn't really make sense, but it's, stimulating the corresponding receptors for you. To see the orange, it would be stimulating the red and a little bit of the green, and then the blue, it's just stimulating the blue. And then, you have people who are green down there. Right? So if you keep on staring at the dot right there, and now I'm going to switch to another picture, and for a moment, you would see color on this picture, right? You would see the green landscape and the blue sky and everything. And after you blink a couple times, you will realize, oh, this picture is actually just black and white, right? So how does this work? Well, it's the same thing, right? By tiring out certain receptors in your vision field, when you switch to another picture that requires their activation, because they have been tired out and they're weaker, they don't register as strong. So the other receptors start to dominate, and then you see colors that are not really there. All right. Oh, okay. I actually already did this question with you. I randomly made up a question, but apparently it was the same question I have for you. Okay. If you switch from a green screen to a yellow screen, the answer is red. Okay. And I already explained why. There you go. Hey, so what about people who are colorblind? Well, color blindness, as you learned from last semester, is an X-linked condition where the person is missing certain pigments, certain molecules that will allow them to detect color in their cone cells. And there is something called the Ishihara test. And if you look at this, you should see the number 74. If you see 74, you don't have red-green color blindness. If you see another number, then you probably have color blindness. And if you didn't already know that, you might want to get it checked out. Okay, so this is a typical picture that you will see from an eye doctor who takes a snapshot of your eye. You can see the optic nerve is right here. That's, is seemingly is where all the blood vessels are coming out from, but this is where the sensory neuron is going to basically exit through the eye and it's going to continue to the brain. And to the side of the optic nerve, you will find the macula. And the macula is basically like this general region that allows you to create sharper image. It's whatever you're focusing on, right? I said. And inside the macular is the fovea with the highest density of cone, and it creates the sharpest image. Now, um, I mean, I don't really know how your setup is at home, okay? Uh, on my screen, I can see the PowerPoint slides taking up most of the screen, and then my video is like on the side of the screen. Okay, just a really small window with me there. If this is similar to your setup, okay, then you can try this activity with me. Okay, I want you to read the words on the slide. Okay, just read them to yourself. And then I'm gonna put up my fingers in the video. Okay, so like you might see my hand waving, right? Don't shift your focus. Keep on reading the words on the screen. You might see my finger moving. Okay, hand moving, but you cannot tell how many fingers I'm holding up. Okay, and that's what I'm trying to say. When you're focusing on the reading, the words are on the fovea. Everything else is on your peripheral vision. Okay? If it's on your peripheral vision, you have an idea of what's going on. Like you can see me waving my arms, but you cannot make out details about the peripheral vision. You cannot see how many fingers I'm holding up. Right? If you shift the focus to my video now, you can see I'm holding up two fingers, then the words on the screen becomes peripheral vision. You know that there are words, but you can't really see what the words are saying. Okay. In other words, you can only focus on one thing at a time. And whatever you're focusing on, that's going to be projected onto your fovea. All right. Same thing, I guess. If you're writing notes as I'm talking to you and you're focusing on writing notes, you can see the writing on your paper. You know there are words on the screen. You can see the words, but not in a sharp image, right? Now, people sometimes get confused between macular and fovea. The fovea is within the macula, right? And the macula is where you produce sharp images, but the sharpest image is going to be in the fovea. As images get further and further away from the macula, they become increasingly lacking in details, right? You still know what's going on, but you won't be able to tell me too many things about it. Okay. Now, the optic nerve exits through the blind spot or the optic disc. Okay, there are no photoreceptors there, so each eye is going to have its own blind spot. Now, if you are doing this on a computer or with your phone, doesn't really matter. You can try to find your blind spot. Okay, so you are going to close your right eye. Okay, so you can cover it up, or you can just close the eyelid. And your left eye should be staring at the plus sign. Okay. Now, if you're staring at the plus sign, you might not see the circle. Okay, because right now at this distance, if I'm staring at the plus sign, I cannot see the circle at all. Then the circle is at your blind spot for your left eye. If you are still able to see the circle, even if you're staring at the plus, then you can move closer to the screen slowly or further away from the screen slowly, and eventually you're going to hit a point where the circle completely disappears. Okay? And that's the blind spot for your left eyeball. Okay? If you're doing it on the screen, you can move the screen instead of moving your head. It might be easier. Okay? Similarly, you can find the blind spot of the right eye, but this time you will cover up your left eye and you'll be staring at the circle. And then you move closer to the screen until the plus disappears. And then you will find the blind spot for your right eye. Each eye has its own blind spot, but if you open both eyes, then they compensate for each other's blind spot. So there is no true blind spot if both of your eyes are open. Okay? The blind spot of your right eye will be covered by your left eye and vice versa. Okay? So we have a pretty decent, 180 view of our surrounding. Okay, so a couple of questions here. I'm just gonna, yeah, question, go ahead.
Um, I'm just a little confused as to where the macula is. So
Yep.
it's in the retina. So is there only like one of it, or is it all along the retina?
Yeah, that's a good question. There's only one of it, okay? The macula. Let me go back to this picture right here. Yeah. So the fovea is a single, it's like a kind of like a dot, right? And then the region around it, that would be the macula. Okay. And it's located next to the blind spot. Okay, next to the blind spot. So each eyeball would have this general region of macula for sharp image, and then within the macula you have the fovea, which creates even sharper image. Okay, it's like HD 720 versus 1080, sort of. Does that answer your question, or you have a follow-up question? Okay, got it. Great. All right, so there we go. The cornea is part of the sclera. Three types of cone cells. You should know this one, right? We talked about it a couple times already. It's red, green, and blue. Which of the following controls the size of the pupil? Maybe we'll do this one. Size of the pupil. We talked about this actually over here, right? Size of the pupil is controlled by the iris. Okay, change in pupil size regulated the amount of light entering the eyes. Okay, so that's the iris, which is C right here. And last but not least, the sharpest image is projected onto the fovea. Fovea. Okay, let's talk about how we change the shape of the lens and how it relates to shifting our focus from looking at things far away and nearby. So lens is a crystalline structure. It's transparent, let light pass through, and it's kind of separate the eye into anterior portion and posterior portion. The shape of the lens is controlled by suspensory ligaments and the ciliary muscle or ciliary body. They mean the same thing, right? So you can see the ligaments all here. Now this is like a side view, right? But if I were to look at it from the front, this would be the lens, and then the ligaments would be all around it, kind of like drawing a sun. And then outside, you would have the muscle layer. Okay, the muscle is like a circular muscle that kind of forms a ring around it. Okay, so this is the muscle, ciliary muscle. And then these are the suspensory ligaments. Now, the ciliary muscle, okay, to watch my hands gesture for a little bit, okay? The ciliary muscle forms a ring. And then when it contracts, it kind of grabs onto each other and then it makes the ring smaller. When it relaxes, then it opens up. Okay, so again, the muscle, when it contracts, it grabs onto each other and it makes the ring smaller. And then when it relaxes, it opens up. Okay. Do I have a rubber band here? I need to have a rubber band to kind of show you this. I suppose I don't have one. Hold on, let me go grab one and then you can draw a little bit, I guess. All right, so the rubber band is like the suspensory ligament. And right now it's loose, there's not a lot of tension in it, right? But like when you pull on it, then it has more tension, right? So remember, this thing is connected to the muscle. So when the muscle contracts, is smaller, then there's not a lot of tension in the suspensory ligament. But if the muscle relaxes and it pulls on all sides, then now because the ring opens up, then there's more tension in the rubber band. So we're going to keep that in mind as we talk about the next concept, which is visual accommodation. Visual accommodation is a automatic process that happens when you are shifting your focus from far away to a nearby object. Okay. An example would be trying to read the bus number as the bus is coming towards you. Okay, so it's really far away and then it gets closer and closer and closer. You have to shift your focus from far away to nearby, right? Or if you think back to like a traditional classroom setting where you have to look up to look at the blackboard and then look down to write your notes, right? Looking up is far away, and then looking down is nearby. And in that process, the shape of your lens has to change in order to help you maintain focus. This is called visual accommodation. So when you're looking at nearby object, the lens is allowed to bulge. Okay, basically it means it has to be thicker. Has to be thicker. On the other hand, if you're looking at far away things, the lens will pull thin. It has to be thinner. Thinner. So how does it happen? Well, let's focus on far away first. If it's far away, we need a thinner lens. So how do you make a thinner lens? You need to pull on the lens on all sides. Right? You need the ligaments to be pulling on the lens on all sides to kind of roll it out, right? So that means the ligaments needs to be tight. Now, how do you make the ligaments tight? You need to make the muscle create a bigger ring, right? Open it up, right? So to open up the muscle needs to be relaxed, right? So muscle relaxed, ligaments are tight, and the lens will be thinner. When you are looking at something that's nearby, the muscle needs to contract. It's contract means they grab on each other. The ring is now smaller, not pulling on the ligament as much. So the ligaments are slack. They have little tension and nothing is pulling on the lens and the lens recoils back into a thicker shape. Okay. As a person ages, the ability to visually accommodate is going to decrease. Okay. And that is why people need reading glasses. Okay. When you try to read like really small writings on like a medicine bottle or something, you can't really see it because basically the suspensory ligament has lost their elasticity, so it takes longer time for them to recoil. Okay, and it takes a long time for your eyes to try to pull back to that thicker shape. And without changing the shape of the lens, it makes it more difficult to maintain focus on what you're reading. So that is called presbyopia. Presbyopia, right? It's the loss of ability to visually accommodate due to aging. Right? So, you just wear reading glasses for it. It hits pretty early, like, early 40s, mid 40s. And, I can't read stuff as close as I used to be anymore because of this. All right. We talked about the aqueous humor, which is at the front right here. That's where you will find it. And then the vitreous humor is going to be at the back. And like I said, the aqueous humor will help nourish the lens, which is right here, the lens as well, as the cornea. And we need to drain it. If we don't drain it, then the pressure will start to build up in the eye, and that pressure will translate to the back and start compressing on your optic nerve. And when that happens, it could lead to glaucoma. Okay, glaucoma is due to increased eye pressure. Increased eye pressure. And at the beginning, the person will start to lose peripheral vision, right? You can still see what you are trying to focus on, but the peripheral vision start to get progressively um, narrower and narrower, and there's no cure for glaucoma. The treatments are aimed at reducing eye pressure. So there are eye drops that you can use, the doctor can give you to reduce the production of the aqueous humor, for example. And there are also medications that will help improve the drainage to slow down the progression of the disease. Okay, floaters. Uh, Anybody have floaters? If you have bad eyesight, you probably have more severe floaters. These are debris that are kind of coming off from your from the retinal layer, okay? And they tend to build up in the vitreous humor, the gel-like substance over here. And so, these things start to flow all around, and then it becomes part of your vision. Okay, when the light ray comes in, and then it hits the fovea at the back. Okay, these floaters are in the way, so you see them. Right? You see them flying around. Okay, and sometimes aging is gonna do that as well. And it's also possible for the retina to kind of detach a little bit from the eye as you age, and that creates flashes. Right? And that's why the eye doctors say if you see flashes and lots of floaters, you need to get it checked out. Because you can have retinal detachment and then you can lose your eyesight from it, and of course that's not good. Okay, but floaters are very common for many people. Like if you look at a bright blue sky and you blink, and then you can see them flying around. Okay, cataracts is another one. Cataracts is basically the lens crystallize and becomes opaque. Okay, opaque means it's the opposite of transparent. Right? Light cannot go through anymore. It's a progressive disease, and then you lose your vision over time, right? Fortunately for cataracts, you can get artificial lens to replace the faulty lens, and then you'll be able to see. You will even be able to see even better afterwards. My mom, as well as my grandmother, they both had cataract surgery, and afterwards, my mom doesn't even have to wear glasses anymore because the lens that she got corrects for her nearsightedness as well. So it's fixable. Right? It's not as bad as a glaucoma. All right, visual pathway um, to the brain. This one is actually quite complicated. So let's do it together. Now, I would ask you to get the diagram from your study guide. But if you don't have this one, it's not too bad. This one you can just draw it quickly. We'll answer the questions in a bit, but let's draw this out first. Hold on a second. I'm gonna copy it over. I'm very scared of the computer freezing again. I don't know what happens. Okay, there we go. It's working. So first of all, what are we looking at? We are looking at the eyeballs, and we're looking at from the top. right? So it's like if you have a transverse section, okay, cut it this way, and then you're looking at it from the top. And these here are the eyeballs, right? Let me just zoom in a little bit right here. This would be the left eyeball, left eye, and this is going to be the right eye. Now, when we look at things, there are things that are on our right side, and then there are things that are on our left side. Things that are on the right side, we say that it is our right field of view. Okay, you have to use color for this, otherwise it will make absolutely no sense. This is the right field of view. Right on the side for you. Field of view. I'm gonna put like a blue person here. Right field of view. And on the other side, we would have the left field of view. Left FOV. Okay, why don't we do the left field of view first? The eyeballs have the pupils at the front. It's more opening for the light to come in, and light can only travel in a straight line, which means the left field of view is going to enter the eyeballs and it's going to fall on the right half of both of your eyeballs. Okay. Left FOV falls on right half of eyeballs. And then the other way around, the right FOV falls on left half of eyeball. Both eyeballs, okay? Do you understand that? Right? Do you understand the right field cannot go to the right half of the eyeball? Because light cannot bend like that, right? Light travels straight. So stuff that are on your right will end up on the left half of the eyeballs, and then vice versa. This is the first thing you have to understand. And if you don't, ask. Okay, this is the pupil. Pupil. All right, so what happens? Over here, this is going to be the left optic nerve. Left optic nerve. It's a bundle of axons. It's outside of the CNS, central nervous system, which is why it's still called a nerve. And you will have information from your left field traveling within it. Okay. Same thing over here. We will have information from the left field traveling here, traveling here, traveling here. And then these information will meet in the middle. This is called the optic chiasma. We labeled it in lecture 6. It's in front of the hypothalamus and the pituitary gland. You can look back at the diagram later on. But this is where the paths are going to cross in the middle. And what happens is, if it is information related to the left field of view, it is going to cross the middle. Okay, so the one from the left eyeball will cross the middle, and the one from the right eyeball will actually just continue on the same side, and then they will go and go and go and go together like that. After the optic chiasma, you're already inside the brain. You're inside the central nervous system, so we no longer call it nerve. It is the right optic tract now. And the tract will take the information to the right thalamus. The thalamus is for filtering information, and that will get projected onto the right occipital lobe. So the entire left field will be processed by the right half of the brain. Similarly, for the right field of view, it will land up on the left half of the eyeball, but this time, the left optic nerve will stay on the left side, whereas the right optic nerve with information from the right field will cross the middle and continue on. So over here, it's the exact same label, but with left instead of right. Okay, left thalamus, and then this is left occipital lobe. So let's take a look at the question. The right optic nerve carries information from which field of view? So right optic nerve is right here, which I forgot to label actually. Right optic nerve. And all you have to do is look at the color. Right? What information does it carry? Is it the right field or left field? It's both red and blue, so it's both. What about the left optic nerve? Same thing, right? Left optic nerve, both colors, both fields. But once you get past the optic chiasma, the crossover point, the nerve becomes the tract. And the right optic tract will now only contains information from the left field. It's only red, right? Whereas the left optic tract will carry information from the right field only. But what about the part of the eye? The right tract contains information from the left field of view, and the left field of view is going to end up on the right half of the eye. Right halves. Similarly, this is going to be left halves. Okay, so it's something that you really need to sit down and draw it out a couple times with the colors and try to make sense of it. Okay, you have to be able to keep track of what information goes. You can feel that this is going to be a confusing question, right, on the test if you are not clear about what goes where. Do you have any questions? Okay, I think I'm gonna stop the eye stuff for now. We'll continue on the Wednesday. But I do want to take a moment to, do maybe do some study guide with you guys on the brain things. Because it was a lot of information, and I'm not sure if you had time to go through it yet. Uh, but I know you have the answers, but then maybe it's a little bit different when we talk about it together. Okay. So here we go. Let's just review some of these things. Okay. Three layers of the meninges from the deepest to the most superficial. So the meninx, which is the singular for meninges, starting from the deepest would be the pia mater. Pia mater. Okay. So it's the deepest layer, and then it is in direct contact with the CNS, with the brain or with the spinal cord. It follows the contours of the CNS closely. So if there is a gyrus, it will, follows the bomb. If there is a sulcus, it will go in, and it's just really a tight adherence to the brain or to the spinal cord. What about the middle layer? Does anybody remember the middle layer of the meninges? Yeah, thank you. This is the arachnoid matter. Arachnoid matter, which is a spider web-like layer. And the arachnoid matter also contains the subarachnoid space. The subarachnoid space is filled with CSF, cerebral spinal fluid. And finally, the most superficial layer is called the dura mater, which translates to tough mother. And dura is really durable, right? This is the outermost layer, and it's in contact with the inside of the skull and spinal cord. Sorry, and backbone. Okay, backbone is called vertebral column. Tough, fibrous layer. Let's not forget, it is actually two layers, but can separate at some points to form dural venous sinus for CSF drainage. Okay. If you haven't been using the study guides for studying, you definitely should consider doing that. Okay, I'll leave that at the top of the screen. Describe how each of the following protects the CNS. So skull and vertebral column, it's pretty straightforward. They provide physical protection against impact and trauma. Like if you physically hit the head, it's okay. It's not gonna, damage your brain necessarily because of the skull. Okay, meninges, uh, again, working with along, I guess we can say along with CSF, cerebrospinal fluid, provides shock absorbance. Shock absorbance. It also offers physical protection. As I said, the dura mater, the outermost layer, is pretty durable, so there is some kind of physical protection right there. Now, blood-brain barrier, just want to remind you, it is formed by astrocytes. Yeah, no, I know. I already posted the answers on the study guide. But like I don't know if people actually use it. So like I'm just using this time to, go through it again to help you refresh your memory. But if you need to go, no problem at all. Okay, if you already know these things and you don't need a refresher, that's okay. Um, Anyways, so this is formed by the astrocytes, which is a type of glial cells, right? There are different types of supporting cells that we mentioned, right? There's the glios, there's the astrocytes that forms the BBB. There is the Schwann cells that forms the myelin, which we might get to in a second. Okay, so the BBB basically helps regulate, regulate contents of the CSF. Okay, it can screen out toxic substances from blood and prevents them from uh, going into the CNS. Right? So it's not a perfect barrier. There are harmful stuff that can pass through. Things like mercury would able to go through, even with like, things like cannabis, for example, like the opioids can go through and cause different effects on the CNS, but nevertheless is a barrier. Okay, cerebrospinal fluid (CSF), they are formed by ependymal cells. Ependymal cells. Okay, so with like I said, there are various glial cells that help support the neurons and protects them. And the ependymal cells are found in the choroid plexus. We wrote all these down. Okay, they are in choroid plexus of each ventricles, each ventricle in the brain. The lateral ventricles is gonna have their own choroid plexus. The third and the fourth ventricles will also have it. And the CSF is gonna provide buoyancy. Right? So your brain is kind of floating. It helps wash away metabolic waste, distribute neurotransmitters, right? So as you are producing the CSF, you need to continuously drain them. Okay? If you don't drain them, they could build up and it could cause problem like this picture. Does anybody remember the name of this situation here? I'll give you a hint. It starts with H. If I show you a picture like this on a test and ask you to type the name, what would you say? That's right. Thank you. It's hydrocephalus. So hydrocephalus is when CSF drainage is blocked. And so what you're looking at here is actually the lateral ventricles, and they're being enlarged because of all the pressure um, pressing on it, right? Can I repeat the last point in CSF? Yeah, this one is distribute neurotransmitters. It's not really a protection of the CNS necessarily, but it's a function associated with it. Okay, so sometimes once you release the neurotransmitters, you need to go to different places of the brain, um, and the CSF it's going to help with that process. Okay, let's do a little bit more. We already did this chart in class. At least I think so. Let me see. Um, yeah, right here, this guy, right? Okay. So we talked about it already. It's the flow of CSF, where they're produced, where do they go, and it's passing through all these different structures along the way. Okay, so I'm not going to do that. 5 different types of glial cells. Yeah, let's do it here. We already mentioned astrocytes. There's a quick question. There's a lot to remember from nervous system. Any idea how to remember properly? I mean, basically what I'm doing right now, right? Like I would strongly encourage you to write down the stuff. Um, so make point form notes and try to recall as much as you can with a piece of paper. And then whatever you don't remember, you can add the details with a different color, and then you study it again. And then you would start the process again from scratch, and this time hopefully you'll be able to fill in more details, and then you just kind of build upon that, right? I mean, different people have different strategies. For me, that's something that really works for me. Like that, I would just whenever I have time, I would just pull out a piece of paper and then start writing down whatever I remember, and over time it kind of builds the memory better, right? Someone suggests cue cards, maybe that's your thing. Cue cards are good, but there's limited amount of information on the cue cards, right? So depends. And also try do the exercise that I provided online. Um, those could be helpful. I know it's a change from what we have, in the first half where you were able to have, access to your notes, but everybody's changed. Like it's a change in department policy, right? So you have to change the way that you prepare for the test as well. Um, sorry. Okay, glial cells. Glial cells, we have the astrocytes. Okay, so astrocytes, main function for the astrocytes is form BBB, blood-brain barrier. But in all fairness, right, when you move on to nursing program, which you are gonna have to take in person, you will have to memorize all these things as well. You will have to take anatomy and physiology when you do the practical nursing. And it's really similar to the stuff that we are doing right now, but like harder, right? So now would be a good time to remember these things. Ependymal cells, Okay, those are important for forming the CSF. We also have the Schwann cells. Schwann cells is to form myelin. And we also have the oligodendrocytes, which also form myelin. Okay, but the oligodendrocytes, they form the myelin in the CNS, whereas the Schwann cells, they form the myelin in the PNS. Okay. CNS is brain, spinal cord, PNS is everything else. Okay, so like the nerve here and the arms, the optic nerve is part of the PNS. Oh, I know it says 5 types, but I think we only did 4 types in class. There is also like microglia that helps with your immune system in the brain. 4, it's fine. Okay, examples of white matter and gray matter. Uh, white matter is primarily for communication, sending signals from one place to another, whereas gray matter is more for integration. Okay. Gray matter is for thinking, if you would. White matter is just to pass the message along. Now, do you remember any white matter at all for all the stuff that we talked about? Like the in the brain. For example, like, do you remember this one? Like if you look at the side, like a mid-sagittal view of the brain, there is a little structure right there on top of the lateral ventricle. That's white matter. Anybody remember the name of that? That's right. That is the corpus callosum. Corpus callosum. Okay, that's white matter. Allows communication between the two halves of the brain. Um, Generally, the inside of the brain is white matter, whereas the outside of spinal cord would be the white matter. So that includes things like the ascending and descending tracts. Okay. The ascending tracts goes up with sensory information to the brain. The descending tracts comes down at the front containing motor instruction, right? Gray matter would be the cerebral cortex, which is like the outside of the brain. And then the butterfly, right? The butterfly in spinal cord. Other gray matter includes the thalamus, even though it's on the inside of the brain, but it is gray matter. And we also talked about the reticular formation. Okay, if you don't remember what the reticular formation is, it's for maintaining mental alertness. Okay, it should be firing pretty rapidly right now, okay, because you are alert and awake. When you're sleeping, it's just kind of dialed down a little bit, but it's still trying to pick up cues and signals from the surrounding in case it needs to wake you up. Right? So if you hear a loud bang on the street, then you will wake up from your sleep. Right? That's the reticular formation um, sending signals to arouse you to like make you like awake. Right? So that's white matter and gray matter. We did this diagram. Example of spinal reflexes. So, like touching a hot surface and retracting the hand. That's a spinal reflex. It only involves the spinal cord. Is really quick. The signal hasn't reached to the get to the brain yet. There is the knee jerk reflex that we talk about, where you tap on the patella tendon underneath your kneecap, and then your knee kick forward a little bit. There is the Babinski reflex. That's the one with the toes. We have a slide on that in the PowerPoint. The toes with fan out for 2-year-old and younger, and it should not be there for adults, right? Cranial reflex involves the brain. So like sneezing, if something tickles your nose and you sneeze, that's a cranial reflex. Coughing, something in your throat, you coughing, trigger you to cough. These are happening in the medulla oblongata. I mean, there are a bunch of them, right? Like, turning your head as a result of like a loud sound, that would be a cranial reflex. Swallowing, vomiting, okay? All these things, cranial reflexes. Okay, how about skill memories? Can you tell me where skill memories are stored? And what are skill memories, anyways? Where do you find what type of information is this, and where do you store them in the brain? Yeah. So skill memories are things that you do with your hands. It's like muscle coordinations. So people say driving, that's right. Uh, typing, biking, shooting a basketball, right? These are skill memories. They are stored in cerebellum, which is the little brain at the back, right? It's very resilient type of memory. You're not gonna forget it. But all the stuff you're trying to study for to remember on the test, that's decorative memory. And these are stored in the hippocampus. Okay. They are screened by amygdala. Amygdala. So if there is no particular emotional attachment to them, it's harder to remember because they get screened out by the amygdala. So examples would be like math formulas, okay? Vocabularies, okay? Factual stuff, right? So those are stored in the hippocampus. It could become long term if you do it again and again and again, but most of it would just be lost if you don't reinforce them. Okay, that's all the time we have for now. Still lots of more questions you can do on your own, and in fact, you definitely need to do that because there's a lot of information to remember. Can I read the circle on the stored in the hippocampus, okay, and then screened as in like filtered by the amygdala. Is that what you need? Okay, so the schedule would be we'll finish the sensory on Wednesday if we have time left. We probably will start the digestive system, And then the following week, we'll finish up digestive system. Hopefully, I'll have a class to review with you on Wednesday, and then the test will be on Friday. Okay. I will give you more information about it later on. Just check the announcement, okay, on Luminate. All right. Probably there will be two recordings for today. Hopefully I'll be able to find the first half. But I don't know. We'll see. Um, that's it, guys. Thank you. Enjoy the rest of the day. See you on Wednesday. Bye bye.